Hi guys, this is Rakesh Gupta, your NEET mentor and motivator. In this video, we are going to talk about, we are going to unbox or review the biotechnology notes that I bought from the Amazon store. These notes has been written by Dr. Neela Bakore, and these notes I'm going to review page by page. But before that, we would be unboxing. So here is the notes that I bought from the Amazon. As you can see, here is the label Neela Bakore Tutorials, and I am just going to unbox this. So let's start our journey, and I will have to break this tape. I have unboxed this very note, and as you can see here, this is the biotechnology notes. Here, what is it? It is written biotechnology notes for medical entrance examinations, biotechnology by Dr. Neela Bakore Ma'am. So in this very section, in this very notes, what you will get is that here it is the front page, which has been very clearly designed, and it is very attractive also. This is the back of this very page, where here it is Neela ma'am and the biography is there, and this is the QR code from where you can access the free videos on the YouTube. So if we talk about the thickness of this very book, this book has negligible thickness. As you can see, there are only few pages inside of this very book. So let me see how many pages are there. So there are only 21 pages. Let me show you. This is page number 21. Only 21 pages are there, and here you can see there is an end to this very book. So 21 pages costed you around 200 rupees, and each unit has an average 300 rupees price for Dr. Neela Bakore notes. So is it justifying, is this note justifying the price that ma'am has decided? In my view, this notes is a quality notes. So don't go for the price of this very notes, because this notes contain a very detailed information. Only point to point information is there, which is exclusively written by Dr. Neela Bakore. Some of my friends believe that this is the handwritten notes. Actually, this is not handwritten. This is a printed notes. As you can see here, if you go inside this very book, you can see the very first page has a printed material, chapter biotechnology and its application, as you can see, and here is the diagrams. The diagrams are very colorful, printed diagrams are there, and here the point wise notes are there. So there are 21 pages, and as you can see, I have shown you the physical cover of this very book, and now we will go inside this very book on my desktop, because I have the PDF copy of this very book, and from there I will show you page by page, topic by topic, what are the topics included in it. Have a look, watch this video till the last. Let's start our journey.
Dear friends, this is the PDF copy of Neela Bakore notes, chapter biotechnology. This is the true Amazon copy, which I am showing on my screen. This is the cover page. Biotechnology notes is here, written by Dr. Neela Bakore. So what are the contents inside it? The first chapter is biotechnology principles and processes, which is from page number 1 to 14, and the second chapter is biotechnology and its application, which is from 15 page number to 21. So there are 21 pages in this very biotechnology notes, as I have already told you in the introduction section. This is the QR code from which you can access YouTube lectures of Neela Bakore tutorials. Coming to our page number 1, here it is the chapter 1, biotechnology principles and processes. The very first paragraph is the introductory part, who gave the term biotechnology, who is the father of biotechnology, and what are the aspects of biotechnology. So there are two aspects, classical and modern aspect, which is given in the very tabular form, point wise it is given, and after that, first recombinant DNA was produced by Cohen and Boyer, and the very colorful diagram is also given, computer generated diagram it is. So this is the page number 1. Coming to page number 2, we have genetic engineering or gene manipulation techniques. So what are the tools which are necessary for genetic engineering, it is discussed here. The very first is enzymes, second is vectors, and the third is competent host. So coming to enzymes, there are various types of enzymes which are used in biotechnological processes. First one is the lysine enzyme, which is used to digest the cell. It means the digest the cell wall, plasma membrane, so that DNA can be isolated from nucleus. So after that, restriction enzyme, which is also known as molecular seizures, their history, who isolated them, and who discovered it, and what are the different types of restriction enzymes, these are also given. And mainly exonuclease and endonuclease type of restriction enzymes, type 1, type 2, type 3 is also given, and a well-to-do labeled diagram is also there. What are the blunt end, sticky end, it has been mentioned here with a very uh, clear diagram. So here is the page number 3. Into page number 3, on the next page number 4, the nomenclature of restriction enzymes is given. What are the steps do we follow before naming the restriction enzymes? And here is the tabular arrangement of all the restriction endonuclease enzymes, their sources and their restriction sites. So some famous examples are given. Here comes to page into page number 4, and on the page number 5, DNA ligase is discussed, which is used to join the DNA fragments. After that, alkaline phosphate, which is used to remove phosphate group from 5' end, so that the reformation of bond doesn't take place. After that, synthesizing enzymes such as polymerase, which helps in PCR reactions, is also discussed. Coming to the next section, cloning vectors is discussed here, and what are the characteristics that a good vector should have, it is mentioned from point number 1 to point number 7. And ori site, that is the origin of replication site, ROP, replication of plasmid protein, selectable marker, cloning site, and recognition site. These all are the necessary features and the characteristics of cloning vectors. It is discussed on page number 5. Coming to page number 6, we have vectors for cloning genes in plants and animals host cells. Types of vectors, if we want to learn, we will be on the page number 7, sorry, page number 6. So the types of vectors is there: plasmid, which is a extra chromosomal DNA molecule which is present in some bacteria. And after that, the first artificial cloning vector, PBR322, which is also mentioned in NCERT, is also been discussed here by Neela ma'am. This comes page number 7, and here is the very basic diagram of NCERT, which is also given and which is very important. After that, another vectors are also there, which has been discussed in point number 3, point number 4, 5, 6, 7, 8, 9, and 10. So page number 7. After that, page number 8 is the competent host cells. What are the features of the host cell? Host cell should possess some characteristics so that DNA transfer technology could take place. After reading these all 3 sections, that is enzymes, vectors, and host, it is mentioned the method of vectorless gene transfer in the host, and what are the processes for recombinant DNA technology is also discussed here. Isolation of genetic material, how does it takes place? what is spooling, and what does restriction enzyme does, and what is gel electrophoresis. This is the very important topic, gel electrophoresis by isoelectric focusing and by density gradient separation. This is discussed in all these points, and this is a very important topic in the view of NEET examination or board examination. After that, transfer assembly is given. The gel electrophoresis or the southern blotting, northern blotting, western blotting, eastern blotting, this all has been discussed here on the page number 10. Coming after that, a polymerase chain reaction, which is also known as gene amplification, is given, and what are the requirements for the polymerase chain reaction, what is the mechanism. This all has been discussed on the page number 11. After that, page number 12 shows a diagrammatic representation of PCR, that is polymerase chain reaction, and it is explained in a very explicit manner. And how does the introduction of recombinant DNA takes place in the host? How is recombinant or transformants identified? So they are identified by some selectable markers. One of the famous selectable markers is the antibiotic resistance gene. And after that, what are the foreign gene products which are produced commercially by biotechnology? We have to also read here. Some bioreactors figures are also there on page number 14. So here is the bioreactor, and we have to learn all the processes for it, downstreaming process. So page number 14. After that, here comes an end to this notes. On the very next page, chapter 2, that is biotechnology and its application, is given, which we would be discussing in the very next video, so that a detailed analysis of this chapter could be carved.
These notes are not affordable to all the students. For this I have my own handwritten notes which I have uploaded on my blog. You can visit this very blog, www.drneelabakore.online, from where you can freely download any chapter notes exclusively written from the YouTube lectures of Dr. Neela Bakore. So please visit this very site I have mentioned in the description, or if you wish to buy this these notes, then the affiliate links are also given in the description. Before going, I would request, please subscribe to my channel. Thank you very much. Be with me. Have a good day.